Hello everyone, I'm happy to welcome you to Immunology lectures. So first I'm starting with MHC class 1 as well as MHC class 2. What about MHC class 1? MHC class 1 is present on all nucleated cells. So red blood cells lack the nucleus and it also lacks MHC class 1. MHC class 2 is restricted to the antigen presenting cells like the dendritic cells, macrophages as well as the B cells. So this is the MHC class 1 and this is MHC class 2. MHC 1 has this component extra if you see here. This is the beta 2 microglobulin. MHC 1 which has this beta 2 microglobulin will interact with CD8 T cells. So this is a CD8 T cell with this T cell receptor. Whereas the MHC class 2 will interact with CD4 T cells. This is the helper T cells, that is a CD4 which will interact with MHC class 2. While MHC class 1, this is a CD8 T cells which are the suppressor cells which interact with the class 1. How do we load the antigens? First, let's start with class 1, MHC class 1. This is how this MHC class 1 looks with its beta 2 microglobulin. The MHC class 1 is produced in this cell organelle, that is the endoplasmic reticulum. The cell organelle along with the MHC class 2, the beta 2 microglobulin is also produced because the beta 2 microglobulin stabilizes the MHC class 1. These are the peptides which are present inside the cell. The peptides can be either the intracellular peptides which are normal to the cell, or it can be pathogens too. These peptides are passed through the proteasome structure, which is also an intracellular structure, and they are shredded. You need to remember this like a paper shredder machine where it shreds it completely. So these big peptides are shredded into small pieces. Now these pieces has to be directed to the endoplasmic reticulum where the MHC along with its component beta 2 microglobulin which stabilizes the MHC are present. So we need a structure which will direct the peptides which are being shredded by the proteasome to the endoplasmic reticulum. What is that structure? That is the TAP protein. The full form is transporter associated with antigen processing. So this TAP protein will take the peptides, or it will direct the smaller peptide pieces which is a product of the proteasome to the endoplasmic reticulum so that it binds with the MHC class 1 and along with the peptide. Now this MHC 1 will from the endoplasmic reticulum will enter into this on the cell surface, thereby waiting for these cytotoxic T cells to recognize it, because this is an MHC class 1 which will combine with the T cell receptor of the cytotoxic T cells, that is the CD8. Let's see about the antigen loading mechanism in MHC class 2. Here in MHC class 2 we have a cytosolic peptide. This is the MHC class 1 which we saw. Here in MHC class 2, similar to class 1, the class 2 is also produced, MHC class 2 is also produced inside the cell. But once the class 2 is produced inside the cell, the peptide place, the place where the peptide, that area where the peptide can attach, that has to be protected, because MHC class 2 is that which is present in the antigen presenting cells. So it is going to process the pathogens and then it has to attach it to that area where the peptide has to be attached. That area should not be occupied by unnecessary peptides or insignificant peptides. Therefore, that area is loaded with invariant chain. We have something known as invariant chain, which will prevent insignificant peptide from attaching to the MHC class 2. Fine. So class 2 alpha and beta chains are assembled in endoplasmic reticulum, but that groove is blocked by invariant chain which are preventing the premature peptide bonding. After this MHC class 2 is complex is trafficked to acidified endosome where the invariant chain is degraded. So that area is where the peptide has to be loaded is empty now, where we are leaving a clip fragment to it. Even now that area is closed with the clip fragment. Even though we have removed the invariant chain, that area is closed by the clip fragment. HLA-DM, another component, will remove the clip, and once the clip is been removed, it is now facilitates a binding of extracellular antigen peptides. This stable peptide MHC complex 2, that is, it has the peptide and it has become stable, translocates to the cell surface. MHC class 2 will we are attracting the CD4 plus T cell recognition. So once it goes to the cell surface, the CD4, it is recognized by the CD4 or the helper T cells. So this is how antigen is loaded in the MHC class 1 2. So MHC class 1 is present on all the cells, all nucleated cells. Any intracellular bacteria or any viral pathogens which are present intracellularly and malignant transformations are displayed by the MHC class 1. But MHC class 2 presents the phagocytosed extracellular pathogens to the CD4 helper T cells, along with the extracellular bacteria, fungal pathogens, as well as the parasitic infections. What is the clinical relevance and the associated drugs? Bortezomib, it is a proteasome inhibitor. As we saw previously, the proteasome is that cellular organelle which will shred the proteins which are present in the pathogens into small pieces so that it can get loaded onto MHC1. If that proteasome is inhibited, then the peptides will not be shredded and there will be no loading onto MHC class 1, right? So proteasome, it is a proteasome inhibitor. Bortezomib, which is a proteasome inhibitor, will significantly reduce MHC class 1 ability to present antigens to the CD8 plus T cells. So this is very important if you are appearing for NBMA exams. But if you take gold salts, which were used historically for rheumatoid arthritis treatment, it inhibits both class 1 as well as class 2 antigen presentation pathways. So now it has only limited use, but this is the mechanism of action for gold salts as well as for bortezomib. With this, we have come to the end of Immunology chapter 1. So I am Dr. Veena. I am a medical doctor. I am anesthesiologist. Teaching is my passion. I love to teach and train young graduates. If you are looking up for a mentor and if you like my videos and like the way I teach, then I am more than open to guide you. So send me a message in the comments. Looking forward to see you succeed. Have a good day.